Mutations lesson. Right now, you should have this Google Slides open on your screen. It should be empty and you should be ready to type in these boxes, which I have overlaid with a text box. So you should just simply have to click on it and type as I type. Hopefully I don't make very many mistakes like I did in my first take, but if I do, I will try to gloss over them as fast as I can. So let's first start at the top by defining what a mutation is. A mutation is any good or bad change in DNA. Okay, I want to focus on a couple things here. A mutation is not always bad. When we say mutation, we think mutant, but it's not always a bad thing. Sometimes that mutation is a favorable thing for the person. Okay, and also the key word right here is DNA. Okay, you have to change the DNA of something to make a change. If I want to change let's just say imaginatively, a green tree into a purple tree, I do not spray paint it purple. Duh. I go into the genes and the DNA of the tree and I change the green gene into a purple gene. So you have to change the DNA of a living thing in order for that change to persist and actually affect the animal. Okay, so these are kind of fun. I'm going to show you what people typically think of when we think about mutations. So starting at the top, these are unusual mutations, but kind of stereotypical in what people think a mutant is. Progeria, accelerated aging. That is a mutation in a person's LMNA gene. What that means, it doesn't matter. All that you know is that this person looks old, but they're about the age 13 because their genes were mutated. Unner-Tan syndrome. This is where the people walk on all fours instead of two legs. So there's a documentary on that. I don't know if that adds credibility to it or not, but it's a mutation nonetheless. Okay, I know this is photoshopped, hypertrichosis, but it is an actual thing where someone excessively produces facial hair. Okay, and it's a mutation. Let's see, this is not a mutation. That's just, if you're curious, that's just an extreme form of HPV, which is a similar virus that causes genital warts. This is being born without an effective immune system. This is a mutation. Oh, we won't stick on this picture for long, but that's uric acid crystals. That's an overproduction of uric acid. That's not blood. This is also something that causes in adults, mostly in their big toe, in that joint between their foot and their big toe. They get all these little crystals and it causes extreme pain called gout. Okay, lobster claw. This is a mutation where some of the genes are flip-flop on a specific chromosome. I believe it's chromosome number 7. It doesn't matter. What's the point? It's a mutation. It's causing all of these problems. The DNA is what's causing this. And this is excessive tissue in the skin forming. I'm assuming some sort of a stop signal is mutated, so the cells just keep overproducing this tissue. This is where the person has a really strange, pungent odor. That's a mutation. Marfan syndrome is when someone lacks connective tissue. So they're very long, kind of lanky, usually very tall people. It was mythed that Lincoln had it, but it's not been confirmed. Okay, so those are your classic mutations. Here are some other mutations that are more common, but we don't think that they're mutations, but they are. Shar-Pei dogs, excessive folds on their skin because they don't receive that stop signal to stop creating that skin tissue. Double-muscled cattle, exact same thing. They don't receive a stop signal to stop making that protein. Extra-toed cats, curly hair, wrinkled peas, cystic fibrosis, red hair, song-learning birds. These are more believable phenotypes that are caused by changing the genes. Back to this. Okay, here we go. So we're going to talk only about point mutations. We'll talk about the other types of mutations in a couple of days. Point mutations are changing a single base in a DNA sequence. Okay, what do I mean by sequence? In that gene, what is the order of adenine, thymine, cytosine, guanine, or what is the order of the ATCGs? Okay, so when I say base, I mean a couple things. They are the exact same thing. A base is the same as a nucleotide, which is the same as A, T, C, or G. Okay, point mutations. The first type of point mutation is a substitution. A substitution is when you exchange one base pair for a different base pair. Again, a base pair would be switching A, T, C, or G, and thus resulting what it pairs with is changed as well. So you switch one for the other. Let me show you this before we type out the 3 different ways this can occur. Okay, so this is what we're looking at is a gene, okay? This is show DNA. Right now I'm going to click transcribe. Okay, it's going to add the complementary base pairs to make the red backboned mRNA recipe code that it's going to send out to the cytoplasm. Okay, now from that transcription, I'm going to press translate. There comes the brown ribosome. There are the green tRNAs. Right here we see the polypeptide, which is your pre-protein forming. It continues adding in the amino acids at the top because it's building that protein codon by codon. Continues to grow. Okay, and it looks like we've reached our stop codon, which is a release factor, and boom, we've got our protein. Okay, I've counted these already. There are 11 amino acids here. Okay, so what I'm going to do, we'll just look right here. It starts with methionine and ends with leucine. Okay, so I'm going to go to edit DNA. And what I'm looking at right here is my original sequence. And what's the ability to type on the dark black right here is my mutated sequence. Right now they're identical. Let's just make a quick mutation to see what happens. I'm going to substitute an adenine for a thymine. I'm going to press apply. Oh, instead of starting with methionine, I've started with leucine. Okay, that's a substitution. I'm not sure if that's going to affect the person in a negative way, but it's still a mutation. Okay, let's put that back. Okay, let's see. Let's just go ahead and delete one of these. Let's see what happens. Look, we significantly shortened this polypeptide or pre-protein. Okay, so that's what I mean by a point mutation. That I did right there was a deletion. That wasn't a substitution. We'll talk about those in a second. But these are point mutations. You change a single point in the DNA. Okay, back here. All right, the first type of substitution, the result would be the lesser of the 3 evils, which is a silent mutation. This is when there is no change made to the resulting amino acid. It's quiet. Most mutations are silent. They have absolutely zero effect on the person who has the mutation. So nothing happens, nothing to talk about. All right, the second of the evils would be something called the missense mutation. Okay, this is when the amino acid is changed. Okay, so you've changed the codon in a way that it delivers a different amino acid. What are some examples of this? Okay, here we go. Curly hair. Changing one single amino acid changes the shape of the hair follicle. So there is a straight-haired follicle. Okay, if you make a missense mutation where an amino acid is changed, you get a sideways hair follicle, and as a result, it produces hair that has some curvature to it. Curly hair, that's all a result of a new amino acid that changed in the keratin 71 gene. All right. Next, let's see. Red hair. Okay, red hair is a mutation. Here's the hair, as you guys can see right there, it's pouring red pigment into it. People that have red hair just don't make the other darker, blacker pigment that we have if we have brown hair or black hair. They just make the red pigment. So as you can see, just the red is dying there. And that's just a single base change to this MCI or 1R gene. Okay. So missense mutations can be detrimental, but just depends on what the missense mutation is. The last one is the worst. This is called a nonsense mutation. And this is when you get a premature stop codon. The protein is not produced or is not as good. Okay, I know that sounds kind of weird, but let me show you what I mean with unicorns. Okay, so a missense mutation. Okay, so we have 3 horses here. I guess they're not unicorns. I would know. I have a 7-year-old. Okay, horses with looks like wings. Okay, in a missense mutation, the purple protein right here is produced, but it's produced differently. Just like you can have straight hair, but differently would be curly hair. So there's my missense mutation. Here, in a nonsense version of the same thing, my protein is produced incorrectly or in lesser amounts. So the wings look just completely weird. And a silent mutation is no change whatsoever. So this is most likely the result of mutation. Is it for it to be silent or missense, or it can be a nonsense mutation. So an example of a nonsense mutation is a really bad disease called cystic fibrosis. Let me show you this picture. These people get very clogged lungs with this mucus because the gene that they produce isn't fully developed because it has a premature stop codon. So instead of liquefying the mucus, it keeps the mucus thick. So these people constantly have to clear their lungs. I know back in the day they used to have to clap on their back to get the mucus out, but it's because their gene doesn't code for a liquefying mucus. It codes for like a thicker mucus. Okay. All right. The next thing that can happen will be something called a frame shift mutation. That sounds scary, but it's not. Okay. This is where it changes the entire mRNA reading frame. Okay. What do we mean by that? Okay. Look at this. A codon reading frame, basically the words are 3-letter words. So every 3 letters or every 3 nucleotides, you get a new codon. So if I were to delete a letter, like let's say I deleted that A right there, all of these bases over here would scoot over one. Or if I added a C here, all the bases over here would move over one, and all of the codons after that would be affected. Okay, so let's see. Let's start with the first possibility. This one will be an insertion of a base to the mRNA. Okay, so think of it like this. Here's your example. This is going to be in non-codon language. The fat cat. Okay? If I added this mutation, I'm going to add an extra T right here. Watch what happens to the reading frame. Okay, I completely shifted all of the letters over. So instead of making sense, you have complete mumbo jumbo. Okay, so that's an insertion of a base pair. No example for that right now. That's understandable. Okay, this one right here is a deletion, okay, which is a deletion of a base to the mRNA. Okay, so let's do our example here. Let's keep it like this. A fat mutation. Okay, I'm going to delete the H in the. Okay, as you guys can see, the entire reading frame has shifted. All right, so I'm trying to go slow, but if you ever need to stop and get caught up on the notes, just press the pause button. I'm going to go to the double-muscled cattle. Okay, this is a mutation that occurs when the gene and the protein is coded for, but the part of the protein that actually stops protein production is not developed. So the animal continues to produce the protein, which is what we eat, but excessively because that protein got cut off too short and it was unable to produce that stop sequence that allowed it to stop. So you get these really, really thick animals. All right, let's go here. Humans, let's see. Maybe you can get different muscles for humans, but I'm pretty sure you can't be doubled muscles for humans. All right, so those are the types of point mutations that I would like you to know. We'll go into the other ones next week on like Down syndrome and other things that can happen when you have extra chromosome material, but this is just the basics. Okay, so what I want you guys to do right now is I want you to go to this interactive. This is where I was just a second ago. So click on that. And if you can, I want you to split screen it because I'm going to have you make 3 changes to this DNA sequence. You can just go to the section where it says edit DNA because I already showed you the transcribe and translate. Okay, so here's your original sequence. Here it is typed again. This is what you're going to mutate. All right, go to this slide right here. Okay, so this was the original sequence. This was the original amino acids. There are 11 of them. I'm just going to type that in. There's 11 amino acids to start with. Okay, I'm going to drag that out just to. Okay, and then that is what the original protein looked like. So when you're comparing your mutants to the original, this is the image that you use. So these boxes are way bigger than they need to be. So I tell you right here in red what I want you to change, and then you just simply answer these questions. How many amino acids is this protein? Okay. Is it more or less compared to the new protein? Are the amino acids the same or are they different? Which type of substitution mutation is this? So is it a silent? Is it a missense? Or is it a nonsense reaction? Okay, that is what I want you to do. So you make the boxed changes. After you make the change though, like let's say I do the first one, I take A and I switch it for whatever it tells me to. I press apply. Let's see. Show protein. All right, before I do the next one, it's important to press the reset button. All right, so please make these changes in the red box. So here you switch the first A for a T. Here you switch the last A for a T. And here you switch this T right there to an A. So this is supposed to say, is this protein more or less amino acids compared to the original, or is it the same amount? Okay, so when you are done with this, I will grade it for participation. Thank you. Is for me to show you that not all mutations are harmful. The whole purpose of mutations is to either give the animal an edge to survive or to create an animal or a version of an animal that is not apt to live in its environment. Basically, it sticks out like a sore thumb and it's more likely to be preyed upon. Okay, so those animals won't survive long enough to pass on their genes to the next generation through reproduction. The ones that receive favorable mutations will. Okay, so let's start with our first of our 3 mutations. Okay, so this mutant will have a large body with long legs, and we will introduce a new predator. So we're going to press begin natural selection.
Okay. 5 generations later.
Okay, let's see. What do you observe about the population of mutants compared to the normal population? Did it increase or decrease in size, and why did that occur? Okay, so apparently the mutants increased in populations throughout the past 5 generations, and the normal animals decreased. It looks like that mutation was favorable. Okay, let's see. With the new predator, let's throw in a small body and white fur. With a new predator. Okay, there's our mutant. Okay, I think we can probably figure out what happens, but let's just look for fun. Sticks out like a sore thumb.
5 generations later.
No more white animal. Okay, what do you observe about this graph? Yep, the mutant population decreased because it was not favorable for the environment. Okay, let's see. A new predator with claws for climbing as a mutation. Let's see. There's our mutant. There are our normal animals.
5 generations later,
Okay, you have more tree climbers compared to normal animals. Why is that? It's more favorable. All right, let's just do one with a new one. Let's see, a large body and long legs with restricted food. All right, there's our mutant. I'm assuming this is our food source. Let's see how the mutant fares compared to the normal one, which is more likely to survive long enough to reproduce and produce more mutants or more normals. All right.
5 generations later,
Okay, let's look at the population graph. Okay, the mutation was favorable because more mutants persisted. All right.